Well, today you get, excuse me, you get to talk and I get to listen. So let's get started. The time is tight to fit all of this into one class period. So there unfortunately will not be time for discussion between talks. We'll have to go straight from the end of one talk to the beginning of the next one. And I will set a timer for 7 minutes. So when 7 minutes have passed, I'll let you know that you have about a minute to finish. Okay, any questions about that? How we will proceed? We will proceed with a change in schedule. Groups 1 and 8 have switched. So group 8 will go first, and then group 1 will be in the group 8 slot later. So the new group 1 is Kiyomi, Kenya, and Justin. Are you ready?
Yep, I think so.
Yes, we're ready.
Okay, proceed.
CFTR mutation in 1303K by Kenya, Kiyomi, and Justin. Cystic fibrosis is an autosomal recessive genetic disease that is caused by mutations or changes in the CFTR gene. This image shows a healthy lung and a lung with cystic fibrosis. CFTR mutation N13OK is a severe mutation that causes pancreatic insufficiencies and severe lung symptoms. The N13OK mutation was identified in the second nucleotide binding fold of the cystic fibrosis gene last year. The effect that N1303K mutant has in the protein structure is that the protein is created, but the absence of amino acid residue leads to the misfolding of the CFTR protein, which is keeping it from moving to the cell surface. To help the CFTR correctly, resuscitation capture or tested capture are used.
Kenya, your voice is breaking up. I'm only catching snatches of what you're saying. So could you check your mic and see if it's plugged in?
I think she's done anyway. For protein functions of mutation N1303K, the protein is destroyed because of the misfolding, and that causes there to be hardly any protein function at all or none at all, shown in this table. The normal CFTR wild type, there is 100% quantity and 100% CFTR function. And in this other mutation, it has 10% quantity or 0 in some cases. And then it has 0.2 or 0.7 percent function. And then in our gene, there is 0.2 to 10.5 plus or minus that for the quantity of the CFTR, and then for the function of CFTR, there's only 0.5% of the whole CFTR function, so practically nothing.
Disease symptoms. So healthy people have an FEV or forced expiratory volume, which is how they measure pulmonary function ranging from 80 to 120. People with N1303K have a range of 30 to 105. Some have normal function while others have serious disability. And air exchange at 30% is significantly lower than normal. That's how it shows it in this table. It shows that lung function for people who do not have CF, cystic fibrosis, under 10 is going to be 61 to 25%. And then 10 to 20, or this is for our mutation. So under 10, it's going to be 61 to 125%. 10 to 20 is 46 to 119%. And then over 20 is going to be 30 to 105%. Whereas in normal, or not normal, but other mutations, under 10 is going to be 68 to 127%. 10 to 20 is 47 to 121%. And then over 20 is 29 to 106%. Pancreatic insufficiency is another symptom that they experience. In N1303K, 95% are pancreatic insufficient, and compared to 85% of cystic fibrosis patients being insufficient. The sweat chloride in a person without cystic fibrosis is less than 30 in all ages. And in N1303K patient, it's 103. That's higher than the average of all cystic fibrosis variants, which is 96. No chloride transport results in heavy mucus and incredibly severe symptoms. That's what it shows in this picture, is how the mucus affects the cells and it sits on top of the cells. 53% of N1303K have pseudomonas, aeruginosa, bacterial lung infections, versus 55% of all cystic fibrosis variants have it. The liver function is not at full function. The liver can't secrete bile or other enzymes. The air exchange because of mucus is restricted and bacteria grows in that mucus. The life expectancy for someone with cystic fibrosis is about 27 years with adequate treatment. But due to the severity of our mutation and the lack of treatment options, it would be understandable if the length of 27 years was cut even shorter. Less than 1% of healthy people experience any of these conditions or symptoms. The treatments, oral pancreatic enzyme supplements, these are taken to help them absorb nutrients and vitamins because their body can't do it on its own. Antibiotics for the lung bacteria, dietary supplements because otherwise they starve because they can't absorb nutrients. And due to the severity of the mutation, treatments are limited, so there's not a lot of options. And that is our presentation.
Oh, yeah, also, that's it.
Okay, you're right on time. So let's end yours and go to the next one.
Sorry, Professor, I'm a little bit late because I clicked on the wrong Zoom meeting link.
Well, I can see you okay. Are you with us?
Yes, I clicked on the right one. I clicked on the wrong one and then I send you an email campus. And then I realized I clicked on the wrong one.
Okay. Okay, so we're at group 3. I should mention that group 2 got split up, so there is no group 2. So we're going to group 3, which is Kaylin, Abby, and Jella. Is any of them here?
Yeah, I'm trying to share
I'm
my
here.
screen right now.
And you can go to the next one.
Is Abby here?
Yeah, you can go there.
Okay.
So Jayla's gonna do the protein structure. I'm gonna do the protein function, and then Caitlin's gonna do symptoms and possible treatments.
For proteins structure, it is the three-dimensional arrangement of the atoms that are in the amino acid chain molecule, the building block, and then also the building blocks all protein, and it's located on the surface of many cells. So by that, on my left, it's the first one is shown on the picture that this is the primary protein structure, and you can see like the amino acid chain. Next.
Mine's on the slides. Mine's the protein function. So just as a review about how the body should function without a mutation and what the CFTR protein is. The normal function of the CFTR protein is the transport of chloride ions across the plasma membrane. And cystic fibrosis transmembrane conductance regulator, also known as the CFTR proteins. They're made up when cells receive instructions from the CFTR gene. When the CFTR genes have mutations, it leads to defects in the proteins. So those who have a decreased stability in the CFTR gene or the CFTR proteins are known to have mutations. Those with cystic fibrosis have little to no CFTR proteins, but able to reach the surface of the cell. So if they do reach the surface of the cell, they don't really function as properly. And then this photo is showing the chloride ions reaching the cell and like going outside of the cell. So this is what leads to, this means that like water or salt has trouble coming in and out of the cell. So that's what's gonna lead to more symptoms like mucus that'll be talked about later. With the D1152H mutation, the chloride ions pass through the membrane less efficiently. So as an example, if you compared the flow of water through a pinched versus a straight straw, you would obviously still be getting the water. It just takes longer and it's more effort. So in this case, the mutation, the ions are still going to the protein, just less efficiently. You can go to the next one. So in this slide, the mutation's a type 4, as well as a missense mutation, meaning that one base pair has changed to alter the protein. Yep. You can go to the next one.
And then some of the symptoms for a mutation are post-nasal drip. There's also a persistent cough with phlegm, slightly higher sweat chloride levels, which can lead to saltier tasting skin. Lung function decreases with age, so as older you get, the less your lungs function properly. You can get repeated lung infections, and then most people with this variant are likely to be pancreatic sufficient, meaning that there is no pancreatic enzyme supplements that are needed. And this image shows what a healthy lung looks like, and then a lung affected by cystic fibrosis with the thickening of mucus. And then some drug treatments that are available are Trikafta, which is an Elexacaftor, Ivacaftor, and Tezacaftor. This drug is really good because it works on 90% of people with cystic fibrosis, and most other drugs cannot do that. This drug is ages 12 and up, and it helps improve lung function and relieves other symptoms. A life expectancy on this drug is possibly like mid-50s. And then another one is Symdeko, which is an ivacaftor and a tezacaftor. It is ages 6 and up, and it improves lung function, and the life expectancy is also about mid-50s. And then there's Kalydeco, which is just an N factor. It is a potentiator, which means that it clears up the lungs. It is ages 4 months and up, so that's really good. And then life expectancy is also mid-50s. And then antibiotics are often used with people with cystic fibrosis to fight lung infections. And then there are also other treatments that have nothing to do with drugs. There is airway clearance, which is high frequency chest wall oscillations, where on the image on the right, you can see the boy wearing the chest and or wearing the vest on his chest, and that vibrates and helps clear up the lungs. And then there's chest physical therapy, which is an airway clearance physical therapy to drain the lungs. And there's huffing, which is a form of coughing to clear the airways, but it is like softer on the lungs than an actual cough. And then infectious control strategies to prevent the spread of bacteria, like hand washing, sanitizing, cleaning, so that there's less likely to catch an infection of the lungs.
So in conclusion, it distributes the flow of the chloride ions, and it's also able to be tested through plasma. And it's about 6% of people with CF that has mutations in the type 4 class, and it was also an image on the previous slide. And medication and therapy to reduce symptoms are available, because there's some medications that might cause these, like side effects and stuff like that. So, or symptoms, should I say. And yeah, I think that's it. That concludes our presentation.
Okay, good. Thank you. You also are right on time. And that brings us to group number 4, Johnson and Taylor.
All right, are you able to see that?
I can see it.
All right, I'm going to be presenting the whole presentation, but Bennett did do half of the assignment since we're a 2-person group. All right, so our mutation is CFTR R117H mutation. In this instance of the mutation, histidine replaces arginine in the amino acid 117 on the CFTR protein. With this mutation, protein structures may remain intact, attaching to the cell membrane, and is capable of folding. It can act just like a normal protein and works perfectly normal, but if paired with another CFTR mutation, the protein structure can be severely damaged. This is classified as an atypical mutation due to its reliance on other CFTR mutations to create a substantial change in structure or function. On the protein structure, it actually changes on the function, it changes the frequency of gating. It may gate normally, meaning that it is chloride sufficient and properly functioning, or it can open in short bursts, causing a chloride deficiency and mucus growth. Paired with other CFTR mutations can be more severe. One such is the F508del mutation, and it can produce 3 different variants varying in severity. And this is what a CFTR mutation or mutated gene looks like. The mucus growth. The F508del variants, there's 3 of them. They're all labeled here. The first one is a defect in the spatial conformation, and it causes degradation of the protein before reaching the cell surface. So combined with the R117H mutation, it causes severe cystic fibrosis and can stop chloride transport almost completely. The second is affects the gating, and it can be decreased by up to 3 times. However, there are various different levels for how gating can be affected. So combined with R117H, it can range from either minor to severe cystic fibrosis. And then the third type results in a reduced half-life of the CFTR protein. It degrades much faster, meaning that it has to produce more. So when combined with the R117H mutation, it can result in minor to mild cystic fibrosis. Symptoms vary depending on the allele pairing. Symptoms range from being asymptomatic, pancreatic sufficient, lung sufficient, typical health.
Smooth, we out of here.
Other symptoms include elevated sweat chloride levels. Didn't really find anything else on that other than since chloride doesn't get used, it just is expelled through sweat glands. Some pancreatic insufficiency can be caused and a decrease in lung function. Its treatments for this specific mutation are as Kalydeco, which affects the gating of the CFTR protein. It is used in people aged 4 months and older who have at least one mutation in their cystic fibrosis gene that is responsive to Kalydeco. It works to help the gate stay open longer, allowing more chloride ions to move into and out of the cell. The movement of chloride ions helps keep a balance of salt and water in the lungs. An important thing to note is that it doesn't actually increase the number of CFTR proteins at the cell surface. It just helps the functionality. If it were to increase the CFTR proteins, then that would require taking up more space on the membrane and using more energy to transmit chloride ions, which could lead to other complications. The most common side effects are headache, upper respiratory tract infection or a common cold, including sore throat, congestion and runny nose, stomach pain, diarrhea, rash, dizziness. These are all really, really bad side effects, but it's not worse than cystic fibrosis. One serious side effect of Kalydeco is that high liver enzymes in the blood have been reported in patients. They'll typically do blood tests to check your liver. When you start, it's every 3 months during your first year and then once a year, depending on your enzyme levels. This could cause pain or discomfort in your upper right stomach area. Jaundice, which is the release of bile into the blood, which causes the yellowing of skin and eyes. Loss of appetite, nausea or vomiting, dark amber colored urine, and blebbing, which is the lack of blood and oxygen into the tissue. Jaundice actually causes a reversible form of blebbing, or is a reversible form of blebbing. And the irreversible form is when enzymes are released by breaking the membrane, as seen right here. And then there's the reversible where they don't actually break the membrane, but create these little like pockets. And that's what happens with jaundice. Other abnormalities of the eye or cataracts have been noted in some children and adolescents receiving Kalydeco, and eye exams may be performed prior. And that's it.
Okay. You also are right on time, so everybody's got it timed perfectly today. So that brings us to group 5. Katrina, Leanne, and Brittany.
Okay, so we had the mutant S549N. So this is just kind of an overview. Obviously, we're going to go over the structure and function, the symptoms, and the treatments for this. Okay, so the effects on protein function. The protein is made and it does actually reach the cell surface. The cell function is affected once the protein reaches the surface. This mutation causes the channel gate not to open. Therefore, the chloride cannot flow in and out of the cell. And as you can see in the diagram, the one on the left is showing a normal functioning gate. The one on the right is with the mutation, which as you can see is not allowing anything to leave the cell. The effects on the protein structure. The structural issue occurs during the protein folding. Mutation affects the cell's function when it reaches the surface and it causes the cell channel not to be able to open. You can see the diagram on the right. If you look from the left corner, going upward and to the left, it shows what would be a normal of the cell process. If you look to the right and down to the bottom, it shows you where the misfolding occurs and how that affects it going down on the class 3, which is a gating mutation.
So different symptoms of the mutation that we have is that the chloride ions, as she mentioned, cannot flow freely into and out of the cells as they should. This causes an imbalance of salt and water, which means that if you cannot get water into your lungs, there is a little alleles that sweep back and forth. If they cannot function properly and there's an imbalance, then it causes sticky mucus in the lungs and lung function is impaired. And this causes a higher chance of pseudomonas and bacteria. It's a pancreatic inefficiency that also can occur. With pancreatic inefficiency, you can have the enzymes that are supposed to break down food do not get carried to the pancreas correctly, which causes you to have weight loss, diarrhea, a sense of fullness, and extreme nausea. A way to diagnose this is through the sweat test, which is when medicine is applied to your arm or thigh to cause you to sweat, and the sweat is collected, and then they test the chlorine within your sweat. This is just a diagram of some of the symptoms. As I said, the mucus can cause a blockage in the air sacs of the lungs. These are the little alveoles. As I was saying, they sweep back and forth. And then this is a blocked pancreatic duct in the pancreas. And this is also another diagram of a blocked lung. This shows a smooth, healthy muscle. This shows what the muscle looks like when it's blocked by mucus.
So there are a few treatments to the mutation, which include Calideco, Simideco, and then oral pancreatic enzyme supplements. So Calideco is an oral medicine that is aimed to help the protein function normally once it actually reaches the surface of the cell, because up until the point where it reaches the membrane, it functions correctly. It just has a gate deficiency, so it will not open, so the ions cannot flow out of the cell. This works for anyone of the ages 4 months and older. So this is normally where they start their medication on. And then Simideco, it works on certain CFTR proteins at the cellular level, like Calideco, where it once the protein reaches the surface, then it starts working and allows the cell to open up. This one is a little bit more complex because it uses both Tescactor and Ivcactor to help the protein work better. So this one works when you're 6 years or older. And then the oral pancreatic enzyme supplements help the body absorb the nutrients and vitamins that are contained in the food we eat, because otherwise we wouldn't be able to get the nutrients and vitamins that we have consumed. So in the next slide shows the effects of Calideco on an enzyme, where it opens the gates so the ions can go through the cell surface, therefore lessening the mucus. And then the next one is our Simideco, which shows what the two enzymes in Simideco that actually help the protein. The Ivcactor helps the protein stay open longer, so it actually opens for the ions to flow through. And then the Tescactor brings more of the CFR proteins to the surface, so they're not just waiting. So therefore, more ions can be transferred out of the cell. So in conclusion, our mutant S549N is a class 3 mutation because it's gating problems where it functions well up until it reaches the surface of the cell where it won't let the ions pass through. It's considered one of the more serious mutations because of the complication in the channel not being able to open. Therefore, the mucus can build up and therefore your lungs and your pancreas don't work correctly. It's diagnosed mostly in younger people and accounts for 6% of the people who have cystic fibrosis. And once again, the major symptoms are lung deficiency and then pancreas insufficiency, where you can't absorb your nutrients. And then we do have 2 possible treatments that you can take. And that's our mutant.
Okay. You're just on time as well. So that brings us to the next group, 6, which is Abby Kotoski.
Okay. No, I clicked the wrong button. All right. Okay, so, and first of all, I'd like to apologize. I have a puppy, so if she starts making noise, I'm sorry. So my project was on the mutation G551D. So first of all, I want to start by doing a quick overview of cystic fibrosis as a whole. So cystic fibrosis is a progressive genetic disease. It is caused by a mutation that affects the CTFR protein. This causes the CTFR protein to be unable to move chloride. The chloride attracts water, which helps thin out mucus that is produced by the body, which is then moved along by cilia. When the chloride cannot pass through the ion channels, the mucus will become thick and sticky because of the lack of water. When the mucus is trapped, it causes germs to build up, which will cause a person with cystic fibrosis to be susceptible to illnesses. So now to move on to my specific mutation. With this mutation, G551D, it is found in the energy transfer. So with this mutation, the CFTR protein, it can move chloride. However, the ion channel doesn't open, much like the last one we just listened to. Patients can be screened for this mutation by using restriction enzyme analysis. Only about 6% of people carry this mutation, and the life expectancy is 25 to 45 years old. There was a lot of different opinions on whether it was 25 or like 45, which is, I know it's a big difference, but there was too many places that said 25 and 45, so I decided to put 25 to 45 years old. This expectancy can be increased with treatment, which I will talk about more in a later slide. So our first image here is the normal, what people without cystic fibrosis, what it looks like. So we have the CFTR protein is created, and then it can be the chloride is transferred to the cell surface, which allows the attraction of water. And with the other image, it shows my mutation, which shows that the chloride channel cannot be opened at all. On to the next slide for protein structure and function. With this mutation, the ion channels are unable to open, so the ATP that would typically open an ion channel is unable to become hydrolyzed, which in order to open the ion channel, ATP needs to be hydrolyzed. So that's the main issue here, is that ATP cannot become hydrolyzed, so the CFTR protein cannot move that chloride through the channel. So with this, it can cause mucus buildup in the lungs, inside of the bronchial tubes. This can cause airflow to be restricted, which causes less oxygen to be able to reach the lungs, which in turn causes shortness of breath, coughing, and dizziness. So as you can see in the image, there's a healthy airway of someone without cystic fibrosis and this mutation. It shows that the lungs are clear, those bronchial tubes are clear and air can move freely. But then when that buildup of mucus, there's a much more narrow space for the air to pass through. So with the pancreas, the mucus will also build up in the pancreatic ducts, which causes digestive enzymes to be unable to reach the stomach in the volume that is necessary for proper digestion, which will lead to malnutrition as the body cannot digest the food properly. So that can cause a lot of issues with people who are trying to just, eat in general. So possible treatments, potentiators can be used to open the channel. The most common one used for this mutation is ivacaftor, which ivacaftor is, it works by being a potentiator for the CTFR protein. It can be used on patients who are 6 years and older. This medication makes it so that the ion channel is able to open. It increases the sweat chloride concentration, the lung and the pancreas function, and there is a reduction in breathing issues. With the use of this medication, it can increase the life expectancy by many years, and will even increase it so much to the point that the life expectancy, that it's close to the same life expectancy of people without cystic fibrosis. The other treatment as well would be dietary supplements, because, as I said before, with the digestive enzymes being unable to reach the stomach properly, people are unable to digest their food, and that causes malnutrition. These dietary supplements can help these people maintain a healthy weight. And then this image here is the chemical compound for ivacaftor. And that is all I have for my presentation. I know I talk really fast, so I'm sorry it went quickly.
Okay, you're done?
Yes.
Yeah, you squeaked in about a minute under the limit, so.
Yep.
You're
I get
okay.
nervous and I talk fast, so my bad.
It happens to.
Apologies.
Okay. Thank you. And then we go to group 7, which is Michael, Brandy, and Mitchell.
All right, can you hear me?
I can hear you.
All right. Good evening, class. My name is Michael.
I'm Brandy.
And my name is Mitchell Toma.
And this is our Cystic Fibrosis Mutant Project on R347P. R347P is a mutation of missteps inside CFTR protein's first membrane span domain, MSD1. And it just a little bit of facts, global frequency of around 0.2% of this mutation. It just happens globally. Mutations in the gene that codes for chloride channel controlled by the cystic fibrosis transmembrane conductance regulator. And this image just shows what would normally happen, gating opening and clear channel. And then somewhere over here is the faulty channel, which we'll get to next. And so R347P's effect on protein structure The shape of the channel blocks out chloride transport for the most part. R347P has a little bit of misfolding happening here on transmembrane helix 6. And about 6%, so I mentioned 0.2% of people globally have the this type of mutation, but that number is 6% of cystic fibrosis patients having a class 4 mutation like R347P. And so because of the way it is structured, the effect on protein function, what happens is that the gate opens regularly and there's a reaction happening, signaling for the gates to open. Everything happens normally, but because of the way the channel is shaped, it reduces chloride transport to the other side, outside of the cell. And this lack of chloride ion movement is called decreased conductance, allowing less chloride to be transported. And what happens when less chloride is transported through the cell, you get sticky mucus and it clogs up the cilia. And about only 30% of chloride gets through the cell, so not very much.
Scientific research. In a research article first published in June 2005 on WileyLibrary.com, a group of scientists states, we report here a group of 19 CF patients with the R347P mutation of German, Bulgarian, Czech, and Slovak origin, including 2 homozygotes. Most patients presented with early disease onset, pancreas insufficiency, and early pulmonary involvement, suggesting that this mutation can lead to a severe course of CF. Next slide. Pancreas insufficiency, or as it is more commonly called, pancreatic exocrine insufficiency, PEI, occurs when the pancreas is unable to produce enough digestive enzymes. If left untreated, it can cause malnutrition along with diarrhea, loss of appetite, vitamin insufficiencies, weight loss, and steatorrhea. PEI is quite common in patients with CF. As mentioned in the study, patients with R347P are also at risk for a more severe form of CF with early pulmonary involvement and symptoms that include recurring lung infections, chronic wheezing, and chronic coughing. Next slide. Treatments include drug therapies for cystic fibrosis and PEI. Trikafta, which is a combination of the drugs elexacaftor, tezacaftor, and ivacaftor, is used to treat CF. It is also effective in patients with R347P. Combining these drug therapies targets the CFTR protein defects caused by CF and R347P. Ivacaftor helps CFTR proteins stay open longer at the cell surface. Elexacaftor and tezacaftor work together by binding to different places on CFTR proteins, which helps more proteins reach the cell surface. A 24-week study of the drug showed that patients experienced improved lung function, fewer pulmonary exacerbations, a decrease in sweat chloride, a reduction in pulmonary symptoms, and increased body mass index, BMI. Pancreatic enzyme therapy is the most effective treatment for PEI. This treatment requires the patient to take a medication called pancreolipase that provides the body with the enzymes that it needs to break down proteins, carbohydrates, and fats during the digestive process. Pancrease and creon are both examples of pancreolipase medications. Both work well in treating PEI caused by cystic fibrosis. In conclusion, 7 to 10% of those living with cystic fibrosis still have no targeted treatment. Fortunately, R347P patients have treatments available to them. Due to today's advanced therapies, of people with CF born between 2015 and 2019, half are predicted to live to 46 years old or more. Treatments like Trikafta And protease ease and in some cases eliminate some of the symptoms associated with R347P, thus providing patients with a better quality of life. And that concludes our presentation.
Okay. You're well under the time limit as well. So we'll go on to group 8, which is originally group 1. Iden, Boshley, and Samira.
All right. Presentation's ready.
Hello everyone, this is our CFTR group project. We're doing the G542X. It's a first class mutant, which means
I can't hear anything. I see your slides, but I can't hear the voice.
I can hear her speaking. She's just really quiet.
Okay.
I'm gonna try
Speak a little
circling.
closer to the mic because I'm not hearing anything.
Can you hear me now? Can you hear me now?
I don't know if you can hear you.
If she's speaking, I can't hear it.
What about now?
Can you hear her now?
No, yeah, I hear that.
Okay. So, okay. Can you hear me now?
Okay. I think mostly you'll just have to do these slides. Is that okay?
I can't hear either.
You can't hear her either. Okay.
No, it's too low.
All right then. So this is our CTFR group project for the mutant G542X. G542X is a class 1 CF mutant. This means that the proteins are entirely absent from the surface of the cell. Normal cell functioning over here, and then on the right side of the screen, I have a class 1. It's the leftmost one on that diagram. Median life expectancy of somebody with CF is 30 years old, but the severity of this decreases that to, on average, 19 years for this specific mutation.
The effects on the protein fraction. There is a difference between a normal protein and a G542X that is known as a nonsense mutation. For normal protein structures, all the protein, including CFTO, is made of the building block called amino acids that links together into a long chain. Each of the 20 amino acids is used at a different position in the long chain. The letter in the chain spell out a stop signal, which allows the cell to know that it have reached the ends of the instructions and can stop making the protein. In the case of G542X mutations, it changed the codon to an early stop signal. Sorry about that. Which make a completely change in the structures of protein, and the ribosome also slip off the incomplete protein on it this way. And here is the picture of a normal protein and a nonsense mutation. The effects on a nonsense mutation on protein function. A nonsense mutation is presented in 10% of patients with CF. It produces a premature terminations codon in FTO RNAs, causing an early terminations of translations and leading to a lack of CFTO function. The G542X is a nonsense mutations and a split mutations. A nonsense mutation contains an early stop signal. Therefore, it stops reading before finish making proteins and causes CFTO protein function to produce. A slight mutation is the ability of the cell to correctly read the instructions for producing CFTO proteins. In the case of G542X, the RNA is unable to build the correct CFTO proteins that can be containing an irrelevant letters or missing relevant one. And this is an example of when the CFTO functions properly and when it's not functioning properly.
All right, so digestive symptoms for this mutation, it's a little bit odd because this mutation causes pancreatic insufficiency, but only when combined with another CF mutation that is responsible for pancreatic insufficiency.
This means that the pancreas does not produce or transport enough enzymes to properly aid digestion, and that results with dietary requirements being more demanding. Additional symptoms of pancreatic insufficiency include difficulty gaining or maintaining healthy weight, abdominal pains and cramps, and frequent bloating and gas. And then here's a diagram showing the enzymes that are directly cut off as a result of pancreatic insufficiency and what parts of your diet they affect. So for the respiratory symptoms, these are common for most forms of CF. Thicker viscous mucus can impair respiratory function. There's a far higher risk of contracting Pseudomonas than a person without CF. Pseudomonobacteria make their home in mucus in the lungs, and these infections cause permanent irreparable lung damage. 56% of patients in the CFTR2 database with this mutation become infected, less than 1% infection rate in people without CF. Limited lung function range compared to a person without CF as well, and the functional range of the lungs only decreases over time. Additionally, sweat chloride content is increased as a direct result of impaired CFTR protein. This does not impact quality of life by itself, but it's a symptom of the fact this is a class 1 and it results in more sweat chloride content on the surface of cells. Here you can see a diagram of somebody's lungs who has CF and somebody who's not a CF carrier. And then this is a diagram detailing the difference in the lung range of somebody without CF and the lung function range of somebody with this CF variant. Digestive treatments for digestive symptoms. People who have the pancreatic insufficiency form of this, they need to take oral pancreatic enzyme supplements in order to have those stand in for the pancreatic enzymes that they lose as a result of their condition. Maintaining a diet high in fat content also helps to combat the difficulty in maintaining weight. As a result of that, fat-soluble vitamins like A, D, E, and K are incredibly important to offset digestive insufficiency. For respiratory symptoms, physiotherapy is needed in order to knock the mucus loose periodically in order to help with airflow. Percussive physiotherapy is what it's referred to. You can see the diagram of it over here. They usually just sit in different positions and sort of like knock with cupped hands on different areas of the chest region. Bronchodilation involves using inhaler medicine in order to dilate the areola, or not the areola, dilate the bronchioles in the lungs, allowing for easier airflow. Antibiotics are needed in the event of infection to mitigate the damage to the lungs, especially important because of how potentially damaging Pseudomonas can be, given that they cause permanent irreparable damage. Lung transplant is needed in cases of extreme degradation to the lungs, and when the degradation is that bad, replacement is the only remedy. So, conclusions related to this: the CFTR mutation G542X does not produce a protein. No protein results in a nonsense mutation. This causes pancreatic insufficiency that also affects the respiratory system, and treatment for this mutation will not stop it from getting worse. People with this mutation live on average 19 years. Again, this is just the diagram showing the difference between a type 1 and every other type. No proteins on the surface of the cell whatsoever. And this is showing the average increase in lifespan of somebody with CF over time. All right, that's it.
Okay. Thank you. That was group 8. So now we go to group 9. Rosemary, Patrick, and Zara.
All right. Just gonna pull up. I'm not starting yet. I have not started the 8 minutes yet. All right. Let's see. Oh, no, I gotta do this. Can you everyone see my screen?
Yes, I can.
Okay. All right, time starts now. Good evening, class. Biology 130. My name is Patrick. In my group is Zara and Rosemary. We're going to be presenting our mutant, which is R553X. Our mutant is a class 1 mutation. The protein synthesis is defective, resulting in no functional CFTR protein being created. What's happening is a stop codon is being substituted for an arginine codon, causing the protein to stop production before it is finished. And I've included some images here of shortened and unstable RNA. On the left-hand side, a normal looking RNA strand there. And on the right-hand side, a shortened, unstable RNA strand. So with normal CFTR function, the proteins create channels across the membrane for chloride to pass through. With our class 1 mutation, CFTR is not synthesized, and that causes chloride to be unable to pass through the membrane. So the protein building instructions contain an early stop codon that causes the production of the CFTR protein to stop prematurely. The CFTR protein normally, the cell thinks it has reached the end of the instructions, but it hits a stop codon instead. So it thinks it needs to stop producing the protein, and it never actually continues making the protein. And now we're going to go into the effect on protein structure.
So for the protein structure, the normal cytosis fibrosis transmembrane regulate protein move to the cell surface and allows the transfer of chloride and water. It's responsible for this balanced chloride and fluids on the cell surface. The absence of this protein includes dysregulation of the service, the nonsense of the or the stop mutants, which is R553X, occurs in the exon 11 of the cystic fibrosis gene. The patient who has these mutants are hydrocytis. Arginine replaced by the unknown amino acid. This mutant produce protein without the regulation domains, which doesn't create no function protein. For this class, early determinant of CFTR protein during the production due to early stop signal. This means after transcription of RNA is produced, which is different and smaller than the normal RNA for the protein. And also the patients who have this mutant don't have this protein on their cell surface, and perhaps the exon 11 according to this gene is skipping once the mutant is present.
And you can see here we've included on the left-hand side some pictures. On the very left is normal CFTR function, and then going from left to right are the different mutants. Our mutant being the mutant number 1, which is on the left-hand side, but it also shows for comparison what the other mutants may look like. There we go. So again, no functional CFTR protein is made. No chloride transport channel. What was that?
Oh, I'm sorry. Hi, sorry. I'm going to be explaining the issues with our mutant with protein function. So because there is no protein function, there is no, the CFTR gene codes, there's the lack of the CFTR gene codes for transporter class protein that conducts chloride ions. Within our mutant, the genes are not expressed. And the channels for chloride, sodium, and water are not functioning, which this would in turn make for normal production secretions within the body, but because we don't have a protein, the chloride that is secreted is not reabsorbed. Insufficient functional proteins at the cell surfaces result in lack of functional chloride channels. These secretions have minimal to no solution, causing dehydrated surfaces within the body. When surfaces are dehydrated, it becomes hard to move these secretions within the body because there is also no signaling for these chloride channels to work. There's lack of secretions within the body. So moving on to chloride production. With a defective CFTR protein, sodium and chloride in water are not reabsorbed, resulting in sweat becoming hypertonic. It's noted that within 1,298 patients, they were abnormal of 102 MEQ per liter. With lung function, thick mucus and dehydrated mucus within the lungs form, and there's not enough fluid to propel mucus, causing infections and oftentimes destruction of lung tissue. Studies have shown that lung function tends to worsen over time. It shows out of 394 patients, 58% have chronic lung issues such as pneumonia. The pancreas.
We're gonna go into treatment now, just to wrap it up. So promising treatments. One of them is an antibiotic named gentamicin. Gentamicin works by stopping the stop codon so that it doesn't actually stop the protein from being made. So this medication is able to prevent that stop codon from stopping the codon where it would have stopped and continue the normal protein synthesis, and can actually, for this class 1 mutation, it can actually allow for functional CFTR protein to be produced after that. Some routes of administration include intravenous administration, which is in the vein, intramuscular into the muscle, or transmucosal. Most commonly, eye drops are used. Can also use a nasal mist. So in conclusion, our mutant R553X is a class 1 mutation of the CFTR gene, or the CFTR protein. It has a stop codon that prematurely stops the synthesis of the protein being made, and so that results in no functional CFTR protein ever reaching the cell membrane. So there's just absolutely no CFTR protein without treatment, and that's our report.
Okay. Thank you. And that brings us to group 10, Caitlin and Sierra.
Can you hear me?
I can hear you.
Hi, class. My name is Sierra, and both Caitlin and I worked on this project together. I'm gonna read the slide set, and she's gonna navigate through it. So our mutant was W1282X, and we just followed the prompts and the questions. And so talking about What is our mutant W1282X in cystic fibrosis? It's a class 1 mutation and is considered one of the most severe in its class. First class mutations such as W1282X tells the cell to stop CFTR production too early, which results in them having no functional CFTR created. It is shown that around 22% of people with cystic fibrosis have some sort of class 1 mutation, not necessarily this one, but a class 1. And the picture right here on the right, we pulled it up just to show that, you know, a normal function where the proteins can go through the Golgi apparatus, and the blockage is represented on the right when proteins can't go through. What effects on protein structure does W1282X bring? Class 1 mutations include no nonsense and splice mutations, which interfere with the production of the CFTR protein. W1282X causes the cellular machinery that converts the initial product of a gene, which is the mRNA, into a functional protein to prematurely stop making the CFTR protein. And then again on the right, we included a diagram just with the full-length RNA on the left, where there's no mutation and everything is flowing smoothly. And we have our mutation on the right, along with 2 other class 1 mutations, or splice mutations with deletions, where the unstable shortened RNA is and not full length. What disease symptoms are caused by cystic fibrosis and our mutant of W1282X? Respiratory signs and symptoms. The thick and sticky mucus associated with cystic fibrosis clogs the tubes that carry air in and out of your lungs. This can cause signs and symptoms such as a persistent cough, thick mucus, wheezing, exercise intolerance, repeated lung infections, inflamed nasal passages, and reoccurring sinusitis. And we also included a picture of just like the x-ray, I guess that would be of a person that does not have cystic fibrosis and their lung capacity, and a person with cystic fibrosis where it looks more closed in and more clogged and blocked for their breathing airways. What disease symptoms are caused by cystic fibrosis and our mutant of W1282X? Digestive signs and symptoms. The thick mucus can block tubes that carry digestive enzymes from your pancreas to your small intestine. Without these digestive enzymes, your intestines aren't able to completely absorb the nutrients in the food you eat. This can cause signs and symptoms such as foul-smelling stools, poor weight gain or growth, and by growth meaning your height, chronic severe constipation. The treatment plans that can help with our mutant W1282X, there are some over-the-counter enzymes that would work. Their potential therapy would be gene therapy, read-through compounds, or ACTs. So patients with this variant will probably need to take oral pancreatic enzyme supplements every day to help with digestive complications. And that's what I meant when I was saying over the counter. Although this mutant is fairly severe, so it's not as effective as other treatments or procedures. So again, reading through the compounds, read-through compounds help the ribosome skip the early stop sequence and read the rest of the information on the mRNA and produce CFTR proteins. Autolurin was one of these compounds that had been previously investigated, but it was stopped due to a phase 3 trial failure. So in that diagram, it's just kind of showing how compounds could somewhat help cystic fibrosis with this mutant. And then we included a slide on CRISPR because ultimately CRISPR would be more effective than over-the-counter or gene therapy. CRISPR-Cas9 is a form of gene therapy that would be considered the best potential option to help someone who has the W1282X mutation or other mutations. CRISPR-Cas9 could be used to seek out, cut, and replace the defective parts of the DNA. The cell would then use the corrected DNA strand to produce a functional CFTR protein, helping in the treatment of cystic fibrosis patients. So just like what we've learned in class, how CRISPR can delete mutations, that would be effective for this mutation. And then before I brought up ACTS, which stands for Airway Clearance Techniques. ACTS are treatments that help people with cystic fibrosis breathe easier. For some people with W1282X, these are some of the only options they have at the moment to seek out some sort of comfort. Some of the techniques are listed below. Coughing, chest physical therapy, postural drainage and percussion, oscillating positive expiratory pressure, high frequency chest wall oscillation, which is also best, autogenic drainage. Current outlook for patients who are living with CF and mutations. The life expectancy of people with cystic fibrosis who are born between 2015 and 2019 and undergo treatment is predicted to be 46 years. However, patients with class 1 mutations are expected to have a shorter lifespan, younger than 20, due to the severity of their mutation and there being no treatments available at this time. In conclusion, our mutant assigned W1282X is a type 1 mutation that results in a non-functional CFTR protein. Patients with a class 1 mutation have shorter lifespans due to the severity of the mutation and the lack of treatment currently available. The only treatments currently available for this mutation are airway clearance techniques, ACTS. The potential use of CRISPR-Cas9 could help with the treatment of this mutation in the future. Thank you.
Okay. Congratulations to you all. We stayed within the time limit. Class period is just now up, so anybody who needs to leave can do so. I want to thank you all for working so hard on this. I can tell from your presentations how much work and organization went into researching and then preparing your slides for presentation. So I really appreciate that. Appreciate your support in getting all these projects underway and completed. And I want to add that if you have any comments or suggestions for either how to do this project or about the class in general, I'd be very happy to receive them. So with that, let me remind you that on a week from today, next Wednesday, will be the final exam, and it will be available midnight to midnight. And I will send you an outline referring to the exam, what it covers and so on. It will be everything from the beginning of the course until the end. There'll be a 2-hour time allotment with one attempt. This is because it's going to be a fairly short, for a 2-hour exam, a fairly short one.
How many questions are in there?
I don't know how many questions because I haven't done the exam yet, but I have it outlined in my mind, and so I know what I'm going to be aiming for. Fairly short, comprehensive, but not terribly in-depth. Comprehensive in terms of subject matter.
So Professor, just to clarify, today is our last Zoom class, or do we have, our final is going to be the same where we just have no class period, or?
Yes, you're right. This is the last Zoom class. Monday is a holiday, and then the finals week starts on June 1st, on Tuesday. So next week is, there are no classes, only finals, and the final for this class will be on Wednesday, June 2nd. No class, no workshops, no office hours.
Thank you for everything, Professor. It's been a great class.
Okay, well, I hope so. I hope you can take something away from it that helps you from here on, developing your careers. And I will, as I said, I'll set there, I've made a recording of this. And so if you want to hear yourself, your group's presentation, you'll be able to do that.